Our next speaker, and the last one today, would be Martin Giurfa, Professor Martin Giurfa, who is a professor of French and Argentinian origin, and he is focusing on neurosciences at Sabatier University of Toulouse. Right now, he is a fellow of the French Academy of Sciences, as well as German and the Royal Belgian one. He received a CNRS Silver Medal and several international awards for his research. He's focusing on perceptual phenomena, learning and memory in insects, mainly honeybees, starting with behavior to different biochemical aspects. He has brought a cognitive dimension to studies on honeybee learning and memory, because his work showed the bees with a new light given their remarkable cognitive possibilities and skills. He was an ERC advanced grant for his work on the neural basis of insect cognition. Ladies and gentlemen, the ERC grant is something extremely difficult to obtain. Congratulations for this, and time to start your presentation.
Thanks a lot. I hope you can hear me. First of all, it's a big pleasure to be here, and I would like to thank the organizers for the honor of being a speaker in this conference. Actually, I would like also to congratulate you for your endurance. I mean, being here on Sunday at 7 o'clock, shows that you have a profound love for bees, the same love I share with you. So what I would like to do today is to tell you about some new light on bee pheromones that we discovered in recent work we were doing. So the first thing we do is pheromone discovered in the 50s approximately, based on the work of a German researcher called Dietrich Schneider. Schneider was interested by this little insect that you can see here, which is the male of the silk moth Bombyx mori. You can see it has huge antennae, and Dietrich Schneider believed that these antennae were used to detect particular odors in the air. And in fact, that was true. What you can see just close to the male is the abdomen of a female. And this female actually is showing a gland that produces sexual pheromone, which is released to the air. And so what happens is that females release the pheromone to the air, the males get activated by these odorants, and then they walk and they go towards the female for sexual reproduction. That was the first observation that led to the idea there are some odorants in the air that help animals to communicate between them. And then came the idea of a pheromone, when the first pheromone was identified. And of course, it was in this little level. So it's very important, because animals speak pheromones today, but few people know that the origin of the concept was in this little insect, the silk moth. And these people that you can see here, Adolf Butenandt, Dietrich Schneider, Karl Ernst Keisling, were the ones who identified the molecules which are in this pheromone. And there are two molecules, actually. There is an alcohol called the bombicol, which is very volatile, and there is an aldehyde called the bombical, which was discovered later. And so there are in a proportion of 10:1. And actually, these were difficult times to work, because you can see here Schneider showing that it had to kill actually 500,000 females just to extract 6.4 milligrams of the first molecule, bombicol. Of course, today, one single moth would be enough to identify the molecules, but long ago, the methods were not as efficient as those we have today. So what are pheromones? What is the standard definition of pheromones? So pheromones actually are chemicals, chemical messengers that are released by animals to the environment by glands that communicate with the environment precisely, and which allow to exchange information between members of the same species. So for instance, the male and the female of the same species, they exchange information, come here, I'm ready to copulate, or things like that. And we know also that pheromones actually are not just one molecule. As you saw in the example of Bombyx morin, the sexual pheromone is made of two molecules. So, and that's generally the case. Most pheromones are composed by several molecules, and the message is actually conveyed by this combination of molecules. And we also know that pheromones release innate responses, like the sexual attraction that we were speaking about. So these responses are predictable, they are innate, they are genetically determined, they do not require learning. This is something automatic, to say in a word. So what about honeybee pheromones? Of course, there's no time to speak about all the pheromones that bees possess, but everyone knows that bees have a rich and sophisticated code for communication in a social context, and pheromones are very important for this communication. There are many glands producing pheromones in honeybees. Here are just some, not all of them. As I say before, these glands have to communicate with the external part of the body so that they can release substances outside. You have the mandibular gland that, for instance, might release 2-heptanone in the workers. You have the Dufour gland, or you might have different kinds of gland, Nasonov gland, which release geraniol, and so on. So bees produce many pheromones that allow communicating between them. But in the time, actually, we were interested by some of the observations we did, which might change actually our perspective on pheromones. Because everyone speaks about pheromones in the context of communication. But our question was different. We actually started expecting that pheromones could have a different role, which might not be necessarily related to communication. Pheromones, we thought, might be doing something else. The question that we wanted to ask is whether pheromones may affect the learning and the memory of honeybees. In other words, when a bee smells a pheromone, of course she gets a message, but perhaps by smelling this odorant, also its learning and memory capacities might be changed so that the animal may learn better or worse depending on the pheromone perceived. So the question is exactly one. Exposure to pheromones change the capacity of honeybees to learn and to make memories, which, as you see, changes the perspective, because I was telling you that pheromones release innate determined behavior, and now we're speaking about learning and memory, which has nothing to do with innate behavior. So the first question we asked, pheromones could change the way the perceived food reward. And there is a way to test that. Basically, if we see this first movie here, you can see a honeybee which is harnessed. It cannot move. But if you touch the antennae of the bee with a sugar water, perhaps you can click on the movie to show it to the audience. You can see actually that the animal will extend the proboscis, the tongue of the animal. So typical test shows you if they like or not the sugar that you are providing to them. So the typical test consists in giving to the bees different concentrations, 0.1%, 0.3%, 1%, 3%, 10%, 30%, and see when they start responding with this extension of their proboscis. So typically what you can see here, for instance, is something like this. You can see here the different concentrations, sucrose concentration in percentage. These are the different concentrations you will give the honey, and you will see when the bees start extending the proboscis. And you see that most bees extend the proboscis for 30%, but few bees extend the proboscis for the very diluted concentrations. There is another way actually to show the same kind of behavior, which is to say, I can compute actually to how many concentrations did a bee respond. I will show you this. But basically here the question was whether exposing the bees to pheromones would change their evaluation of reward. They decide or not to respond to sugar, and does it actually exposing to pheromone change their decision to respond or not to sugar? And so we expose bees to two kinds of pheromones. It's a negative pheromone because actually it is a deterrent pheromone. Bees are used to actually deter enemies or at the hive entrance. We use also a positive pheromone, which is geraniol, as most of you know. It's an attractant, actually. So we had these two pheromones with different valence, negative and positive. And so what we did is we put the bees during 15 minutes in a little tube exposed to the pheromone. As you can see, here's the bee in the little tube, and then there is a filter paper with the pheromone, and the bee stays there for a while with mineral oil, this is a control, no other, or with geraniol, the attractive pheromone, or with eptanone, the aversive pheromone. And then you leave them for a while, then you take them out, you wait also for a while, there is no pheromone there in the environment anymore, and then you ask them, okay, so how do you respond to sugar? And what you can see here is the following. You can see that there is oil response. You can see the black squares, oil, means the bees that have not any odorant. And you can see that they respond as predicted. The more concentrated, the more bees respond. But you can see also something very interesting. When bees were exposed to the attractive pheromone, geraniol, you can see that the responses increase. So they like now concentrations that they consider to be bad before. And they were exposed to the bad pheromone, to eptanone, for instance, they go down, which means they don't like to respond to concentrations. So you can, as I tell you, you can show the same data in this way. Look at this, for instance. Come on, move on. Okay, so here you have 4 bees, for instance, and you have the different concentrations. And you can see, for instance, bee number 1 that responded just to the last concentration, and she gets a score of 1 because it responded just to 1 concentration. Bee number 2 responded to 3 concentrations, it gets a score of 3, and so on. Okay, so you can give a score to A, and then you can represent the scores, the average scores of your bees, and then what you get is something like this. Let me show you this. Here is it. You can see that the bees that were exposed to oil, no other exposure, they respond in average to 3 or 2 concentrations, but the bees that were exposed to geraniol, the attractant concentration, increased their response to reward. They are more responding to reward now, while bees that were exposed to the aversive pheromone decreased their response. They don't like now, or they like less the sugar compared to the control bees. So then we say, okay, this is very interesting because actually it shows that pheromones change the way the animals feel the reward. It's very interesting, but let's prove that if it is the same for any kind of response. So we say, okay, we were testing sensitivity to reward. What happens with sensitivity to punishment? There is another way that you can test the sensitivity to punishment. And here you have again a bee that is immobilized. Perhaps if you can see this little movie here, you can see the head of the bee just in front of you, and you can see the abdomen. And you might deliver a very, very, very mild electric shock. And then the response, of course, extension of the sting. Here you see it. You see the bee extending the sting as a response to punishment. So what you can do is the same as before with the sugar. You can see that if you give, for instance, different kind of voltages, 0.2, 0.5, 1, 2, 4, 8, okay, some bees respond more, some bees respond less, and so on. And here the question is how those exposure to pheromone change the sensitivity to punishment. And here are the results. So we expose again the bees to the negative pheromone and to the positive pheromone. So now, before, this was more related to food, but now this might be more related to punishment. Let's keep that in mind. So, and in the 2-eptanone more related to punishment and the geraniol more related to food. So we did the same. We put it in the little tubes with the pheromone. We let them there for a while. Then we took them away. We let them resting for a while. And then we ask, how do you respond to punishment after being exposed to the control mineral oil, to geraniol, or to 2-eptanone? And this is what we got. So you can see on the left exposure to 2-heptanone. And now you can see in the orange curve that the extension of the sting increase. Now they are responding much more to the voltage, to the electric shock, after perceiving the negative pheromone. On the contrary, after perceiving the food-related pheromone, or the attractive pheromone, if you want, now respond less to the electric shock, the green curve you can see under the geraniol label. So this is very interesting, and you can show also the same score. You can do it the same kind of calculation. You have a bee, for instance, that responds just to the last voltage and gets a score of 1, a bee that responds to 2 or 3 voltage gets a score of 3, and so on and so on. And if you represent these scores, this is what you get. Here it is. Come on. Here you see mineral oil in the middle. It's the control, means that bees respond to 2 or 3 voltages. But when you expose to geraniol, it goes down. They respond even less. But when you respond to the negative pheromone, they increase and respond much more to the punishment. So these results are quite interesting, actually, at least to us, because they show that the way a bee responds to food or the way a bee responds to punishment is not fixed, and it can be changed by the pheromones according to their valence. So positive pheromones increase the response to food, negative pheromones decrease them, on the contrary, in the case of punishment. So this is the conclusion of this, but then we say, okay, if the pheromones change the response to reward, then they may also change the way the bees learn about reward. And so we did an experiment in which we wanted to know if exposure bees to pheromones will change their learning and memory capacity, not just the way they feel, but also their capacity to learn, to make associations. So what we did is to put our bees in front of this machine, and you can see here in the picture, which is sending constant clean air to the animal, and you can introduce there a pulse of odorant, an odorant. And this odorant is neutral to the bee. The bee does not know this odorant. And then, for instance, you can see here the machine, and you see the bee in a little tube in front of the machine. Then the experimenter will go with a toothpick and touch the antennae with sugar. So the animal will make the association, anytime the odor arrives, then I get sugar. So if the bee learns, the bee will learn to extend the proboscis to the odor, because odor means sugar. Can we activate the first movie, please? So in this movie, you will see a bee that actually is getting the sugar, and now you can see, for instance, it gets the reward. And in the next movie, can you please show the movie? Actually, it might be probably okay. In this case, you see the bee, it cannot move, of course. You will see the red square, actually, and now the bee responds to the odor, even in the absence of sugar, because odor means sugar for this bee. So you can see it responded to the odor, and the odor was indicated by the red square. So this is a very simple way and practical way to see that bees can learn and memorize that odors mean food. And this can be done easily in the laboratory. And they learn very fast, actually, 2 or 3 times odor and sugar. That allows to form very distant memory that they can recover several days later. So what we did here is an experiment in which we use 2 odors. One, which is in blue here, which is associated with food, and another one in black, which is associated with nothing. So the bees had to learn that there is reward, and there is someone speaking at the same time, sorry. Okay, so I was telling you, so 2 odorants, one odor in blue associated with reward, the other odor in black without nothing. So bees have to learn to make the difference, or the other punished. And you test later. We can test, as you can see, 2 hours later, 24 hours later, or 3 days later, because they have a very good memory. And here the question is the same. Okay, we know they will learn to make the difference between these odors. But again, will the pheromones change the way they learn, the we form the memories. So this is what we did. We did the training, but before the training, as you can see, we exposed them either to mineral oil, the control, or to geraniol, the positive pheromone, or to 2-heptanone, the negative pheromone. And then we test them either 2 hours later, 24 hours later, or 72 hours later. So this is what we got. Look, this is amazing, because actually you can see in gray, actually the bees responded to the mineral oil, and you can see that they learned to respond to the rewarded odor. And if you look well, you will see a gray curve at the basis, which is the non-rewarded odorant. But now look in blue, the bees, they have better learning. They are above. And the bees exposed to 2-heptanone, the negative pheromone, they have worse learning. They are below the controls. So again, the pheromones change the way they learn. Not only that, now look on the right panel, the memory test. You see the memory at 2 hours, 24 hours, and 72 hours. In gray, the control groups. They have memory, of course, but look, the memory of the bees exposed to geraniol in blue is much better anytime. And the memory of the bees exposed to 2-heptanone is worse anytime. So the conclusions of this experiment, and that's the end probably or practically the end, is that pheromones are much more than just chemical messengers. They change the capacity to learn and to make memories because they change the sensitivity to reward and punishment. And that is a fully new perspective about the way we see pheromones, and that we can use in a way to improve the activity of this, because the pheromones somehow prepare the bees. If you see, if they perceive an appetitive pheromone, okay, that prepares them to learn better about food. That's the way it works, to make better memories and so on. So what do the pheromones change in the bee brain when they perceive them? Some people might say, okay, perhaps they change the way they smell. That is exactly what we did, and we have the way to look into this little brain of honeybees and record the activity of the neurons that respond to the odor. And basically, the answer is that the pheromones do not change the way the bees smell. We look at this area in the bee brain in blue, which is the area of the bee where neurons responsible for encoding odorants are located. And we can see this region very well defined, and we can see the neural activity there. And to make the long story short, I can tell you that before, for instance, you see two odors, hexanol and linalol, then you have the pheromone exposure in the middle, and nothing changed. I mean, the exposure to, this is actually in the movie you are seeing how these regions change when you expose to odors. But here nothing changed. So the pheromones do not change the way the bee smells. The pheromones change actually the neurons that respond to sugar and the neurons that respond to punishment. And this is why this is the reason why the bees actually learn better or learn worse depending on the pheromone. So I'm finishing here. What I to do is to tell you about some of our new results, telling that pheromones, of course, they help honeybee communication and they are essential for the communication within the hive, but they are much more, helping them to be actually prepared to learn in the right way. Okay, if there is an aversive pheromone 2-heptanone in the environment, it makes no sense to concentrate about food. You should be better in learning about punishment. If there is a pheromone indicating the presence of food, then you should be able to learn in a better way about food and not about aversive events in the environment. That's the message. And I just want to finish by thanking all the people who work with me in Toulouse and support this kind of research. Thanks for your patience and attention, and endurance above all.
And so do we. Thank you. Thank you for embarking on a very interesting subject. In the meantime, some questions have been asked. We will ask them to you. And yes, thank you very much. The first question is, what is going to be the next limit of knowledge about the skills of honeybees? Everything you've said has been fascinating. Where do we go from here? We'd like to know a bit more about that. Where is the next frontier?
Okay, there is something we have to say. I don't know if someone say that yesterday, but yesterday was a very important day for bee people because it was the 135th anniversary of Carl von Frisch. Okay, very famous scientist, Nobel Prize, who discovered the bee dances and many other things about behavior. And Carl von Frisch said something that somehow is the answer to this question. Carl von Frisch say the honeybee is like a magic well. The more you draw water from it, the more there is water to draw. And that's exactly what I feel about bees. There's no limit. Every time we're discovering new things, and every time it's becoming more and more fascinating.
Thank you very much. I believe that this is what we all believe. Perhaps another question, which I personally find very interesting: to what degree a bee can be aware of its neurological and physiological states? Can it consciously, for example, feel a state of pleasure? And if so, do they have their psychoactive substances they prefer, like some substances in nectar, for example, caffeine or nicotine? And do they have pleasure, or do they feel pleasure after consuming those?
That's a fascinating question and a very important one, because if we start speaking about consciousness or things like that, then we have, and many people will have to change the way they consider insects, bees. And it's very important because it also implies that we change our relationship with the environment. We need to consider these animals in a different way and treat the environment in a different way. Concerning the question, yes, I mean, there are some experiments that indicate the presence of some levels of consciousness in honeybees. For instance, I mean, there is something called the metacognition, which is actually what I know from what I know. For instance, if someone would ask me, what is the probability that you get right answering me what was the result of the final game in World Cup 1978? I would say, well, I know 100% right. I can tell you the result right now. Many people would say, no, 0%. Other people would say, perhaps 50%. So what people are doing, they are making some introspective knowledge of what they know. So they say, the knowledge about what I know, I need to be aware, to be conscious of what I know. And you can test that in animals. You can provide an animal with a difficult task and tell him, okay, if you go and you get it wrong, you get punishment. So it's your decision. You go and make the task, or you might decide to leave, which is actually like the students who decide not to go to the university to make an exam because they consider that they will fail. Exactly the same. And when this experiment was done with bees, when the discrimination was very difficult and there was a exit in the apparatus, most of the bees decide to leave, like the students that do not want to take the exam. So what does it mean? Does it mean that they are somehow conscious of what they know and what they can? We need more experiments to answer the question. But you see that there is something there that needs to be explored. Concerning the other part of the question, where they or pain or things like that, it's very difficult in animals to answer these questions because animals do not speak, obviously. But there are ways to address the question. One is what was mentioned in the question. There are different kinds of molecules and neurotransmitters that are in our physiology when we feel pain, and that are released and so on. And you might want to know if the bees have the same or similar ones. And for instance, we found some molecules that could be actually similar to some molecules that we release when we feel pain. It's not proven, it doesn't mean anything so far, but this is a way to try to answer the problem. What is clear is that they like to find some substances like caffeine and things like that in the nectar. Does it mean that these substances provide pleasure? We don't know. We have to be cautious. But I mean, what should be clear in any case, even if we don't have all the answers, is that we cannot consider these little bees as little machines. They are much more than that. They are creatures actually which learn, which memorize, that can solve very complicated problems, that can have perhaps some levels, basic levels of consciousness and so on. So we need to start looking at them in a very different way, which means we need to treat them differently in terms of what we do. It's very important.
Bardzo dziękuję za tą odpowiedź.
Thank you very much for that answer. And another question. Can the bees recognize the smell of the beekeeper, or do they live too short to recognize him? And can beekeepers, for example, recognize the face, the physiognomy of a beekeeper?
Okay, these are very interesting question again. If the beekeeper is using a constant, I don't know, aftershave or a constant odor, it doesn't change it anytime because we are aware we are changing our odorants on the basis of the kind of, I don't know, soap or things we use. But if we are rather constant in terms of our odors, my prediction is that bees can definitely learn the odorants of the beekeeper because they are very good at learning odors, as you can see, as you probably saw in my presentation. So I think, yes, they can definitely learn the odorants. Learning the face is something more complicated. First of all, because remember that many beekeepers use protection, so they might not see the face behind these protections. Others do not use. Okay. What about those? I mean, it's more complicated because they are very good at learning visual stimuli, but mostly, at least this is what we have studied, they learn this visual stimuli in association with reward. In other words, if you tell me that you take a picture of yourself and you put it in some place and you give a drop of sugar water in the middle of your picture, they will learn to go there and they will learn that picture. Does it mean that they would know that this is you? Probably not. Probably this is just a strange flower in which they get in reward. So we need to be cautious. I would not say that they learn faces because it has not been properly proved. The experiments, and we did some experiments in that way, but it's in the way I mentioned. So with pictures, black and white pictures of different faces on which we put sugar water. But this is not people. It's just a picture. And so the bee can just imagine that this is a strange flower having a drop of sugar water in the middle, not a real face. So I believe that they can learn the odors if the odors are constant and do not change in a person. I'm a little bit more cautious and skeptic about learning the face as a whole.
Thank you very much for that answer. And another question, and it's the last one now. Can bees share the information they have remembered thanks to pheromones to other individuals or to other generations? In other words, is the message of culture transmission of information possible?
Sorry, I just heard the Polish version of what you were saying. I didn't get a translation.
Can you hear me? I'm the interpreter working on the English channel.
I have no...
Can you hear me? I'm on the English channel.
Yes.
Can bees transfer the information they have remembered thanks to pheromones to other individuals or to the following generations? In other words, is the mechanism of culture sharing information possible?
Oh, yeah. I mean, in honeybees, there are no real convincing experiments about cultural transmission. But there are experiments on bumblebees. Bumblebees easy to keep in the laboratory because you have these little boxes that you buy. It's very practical. And many scientists work with bumblebees. So I have a good friend and colleague in London, Lars Schitka, and we did our study together back in Berlin a long time ago. And he was studying cultural transmission in bumblebees. And actually, what he was doing is a very funny experiment in which he was teaching bees to he had a disc and he had a sugar drop on it. But he placed actually a glass on top of it, so bees could not reach the sugar. But he had a little nylon thread connected so that bees, if the bees learned to take the thread and move it away, they could get the disc and get the sugar. So, and amazingly, the bees learned the task. So they were learning to pull actually the nylon to get the color disc and the sugar on top of it. And the interesting thing is that he allowed other bumblebees to watch, just to watch what the others were doing. And these guys did it from the very beginning. So showing that they learned by observation. And then he started looking how this information spread in the population of bumblebees, and it went faster and faster, so which looks like cultural transmission.
Thank you very much for the answers. This has been really cherry on top of our cake. A very interesting question, a very interesting lecture. We could be going on and on about that. Nature and insects can be a pleasant surprise, and I believe there's plenty to discover. And still, I don't believe we'll manage to discover everything, because discovering one thing at some level, we realize that there are more levels to discover that are hidden within. Thank you very much for your lecture, sir, once again.